Stanford University.
Thanks, Sally. And actually, Sally's responsible for me being in the Bay Area. So if anybody you have problems with that. Sally hired me originally 12 years ago, 13 years ago. So what I'd like to talk to you about is the work that we've been doing at Berkeley Lab on the MC252 oil leak. And this is a large team of scientists, postdocs, RAs, TAs, graduate students, and even undergraduates. The original science paper that we had come out in, on August 24th had 31 co-authors. So we believe in giving everybody credit. It was a major effort. We are funded through a contract with the University of California at Berkeley from the Energy Biosciences Institute. And just for full disclosure, that institute is funded by BP, and the funding came from a grant, a $500 million grant that the University of California at Berkeley managed 4 years ago. We have a program in microbial enhanced hydrocarbon and oil recovery that was funded 2 years ago, and that's partly why we were able to immediately mobilize and go down to the Gulf and start doing basic research. Because of the conflicts of interest and things that DOE will not let us work to, this is completely unrestricted work. A lot of people have that question, so I'll answer it right up front. It was completely unrestricted, and in fact, BP didn't even see the data before it was published in Science. So they got it the same time that all the rest of you got it. So some people have said, well, just because it was BP funding, you're tainted. I would argue with that. But just for full disclosure, let me get to it now. You will see the Deepwater Horizon spill mentioned. You will see MC252, which is Mississippi Canyon 252, and that is the particular section and parcel for where the well was. You will also see Macondo well. All of those are the same thing. So on April 20th, the Deepwater Horizon, which was a drilling rig, for unknown reasons, but we think it was probably due to rising and rapidly expanding methane. And the Deepwater Horizon Commission report just came out this afternoon. So you can go online and get that. The floating rig burned for 2 days, killing 11 people, and then sank, sharing the 21-inch riser some 600 feet above the blowout prevention device. So actually, that was just a dent. Most of the riser went to the floor, and the bulk of it was coming out one end. There were 3 breaks in the riser pipe. Originally, NOAA placed the amount of oil coming out as 5,000 barrels. That was a rough estimate from surface calculations that they did, and they quickly revised that. Later estimates were 60,000 barrels a day, and that basically has held true. 54.1 million cubic feet of natural gas were being flared, a lot of volatile components. 4.9 million barrels were released to the Gulf, and this is according to the final report from the joint government report that came out on December 17th. The spill has surpassed the Exxon Valdez spill, which was only 0.26 million barrels in 1989, and even surpassed the second largest blowout in the world, which was the Ixtoc 1, 3.3 million barrels, and has now become the second largest. The first largest is actually from the Gulf War. This is the biggest in the USA. The well was capped beginning July 12th. There was no further oil input to the Gulf after July 15th, and the government declared the well dead from the relief wells on September 19th. This gives you an idea of the scenario. Burning rig went to the bottom, and this is 5,000 feet deep, or about 1,500 meters. Originally, you can see the 3 leaks here. They started immediately drilling a relief well. They brought in ROVs to start looking at what was going on, and developed basically a containment box. That didn't work. They then, basically it was clogged by methane hydrates. And BP prepared a smaller containment dome, and then they never basically used that. They also inserted a siphon tube into the riser. First time they did it, didn't work. Second time they did it, they actually started collecting oil. They then started drilling a second relief well, and did a number of operations, including the top kill, where they injected tremendous amounts of drilling mud and junk, trying to kill things within the blowout prevention device. They finally went in and cut off the riser, and then started working on a cap to contain it. And so by July 15th, they had this certified that it was actually containing and it was going to hold, and the containment cap. So on July 15th, there was no further well going in. So I think one of the interesting things is here that 11 people were killed, and we don't hear about that a lot. It's interesting that culturally these workers on these drill rigs are from an international community. They're from all over the world, and not a local community. I think we would have heard a lot more about it, and a lot more about their families and that sort of thing if it had been like a mining community and things like that. So I think we don't hear enough about the 11 people who were killed and the effects on their families. Whoops. This is just to give you an idea of what the oil is like. It's a light crude oil. You can see the specific gravity here. It's typical of the Gulf of Mexico and oil reservoirs that are greater than 6,000 meters. About 35% of it evaporates within 2 days. And in fact, 45% will evaporate within 2 weeks. And here you can see a light crude versus an Oklahoma heavy crude. And this is also similar to what was in the Exxon Valdez spill. So quite a bit of difference, not a lot of heavy fractions here. In terms of concentration, a lot of light fractions, and so that's one of the reasons that it is quite valuable. And this is even more valuable now that the well is dead. This is Macondo source well oil, and you can see how light that is and brownish in color. This is weathered oil, Macondo oil, and you can see once it's weathered and all those volatile components are out of there, it's more like motor oil. So I'll pass this around so you can all see that and get an idea of what the oil is actually like. So what happened? Well, they cut off the riser, as I said. Jetting created small droplets. If those droplets were 10 to 60 microns, they moved out in a deep water plume. Concentrations were fairly low. Highest concentration 10 ppm, average concentration a little bit higher than 1 ppm. You actually couldn't see it or smell it. And a lot of people don't realize that. Also, droplets that are larger than that rose to the surface. And this was one of the problems. Of course, oil then emulsified, and there was various types of cleanup operations in terms of burning, skimming, and they were doing a lot of leak operations. At one time, there was 7,000 ships out there. Tar balls went to shore for the material that did get to shore, and they put in booms. They were largely ineffective. Skimming operations were pretty good. But one of the things I want to emphasize is that for the first time, Corexit or a dispersant was injected at the wellhead or at the top of the blowout prevention device. This caused the droplets, more of the droplets, to move out farther and overall to move out farther. So the basic reason that BP was doing the Corexit injection down deep was not for any other reason other than safety. They wanted to make sure that all of this volatile and heavily flammable stuff was not coming to the surface where all these leak operations were and where they were trying to close the well. Within 4 hours of starting the Corexit injection, a significant amount of the oil moved farther away. And every time that they stopped the Corexit injection, which happened several times, 4 hours later, they were seeing more oil at the surface. So it was primarily a safety issue for BP. Now, 31,800 people were deployed, 4.9 million barrels, and this is from the latest report that came out from the OSAT, and 820,000 or 70% captured, naturally dispersed, chemically dispersed. A lot of it evaporated, and of course, 23% was remaining or probably biodegraded. There was 441 controlled burns. They used 1.84 million gallons of dispersant. That's the most that's ever been used in any site before. About 1 million down deep, and slightly less than that at the surface. So what we wanted to do was to use a systems biology approach and basically look at geochemical processes, microbial population correlates with the geochemistry, measurement of biomolecular activities during processes, simplified laboratory communities, cellular network analysis of function, molecular analysis, and then also predictive models of cellular function, predictive models of community function, and finally predictive models of geochemistry and ecology. To do that, we basically had 3 major areas we were looking at. We're using microbial community structure and function. With in situ analyses, we used the Berkeley phylochip, the GeoChip, phospholipid analysis, and I'll talk about all these later. Metagenomes, metagenomics, and metatranscriptomics. And then we did isolation and lab simulations using the synchrotron advanced light source, FTIR real-time analysis. High pressure simulations, microrespirometers, so rates of biodegradation, phenotypic microarrays, so rapid biochemical characterization and biodegradation of growth curves, and then the geochemistry for linking the structure and function. So we use stable isotope analysis of petroleum daughter products and petroleum, and then also linking that with bioavailability, coupling that with community and populations, and then trace metals, hydrocarbons, and limiting nutrients, nitrogen, phosphorus, that sort of thing. So all those things simultaneously. So it was a scientific or a scientist's fantasy or dream in some ways that we got to do all this. We were on 4 different ships, and we were out there from May 25th to October 20th. So unlike a lot of the other groups, we were out there continuously until October 20th and collecting samples that whole time. We initially were on the Ocean Veritas and the Brooks McCall simultaneously. We then switched on to the Ferrel, and then finally onto the Gyre. And the Gyre, we were primarily doing sediment analysis. We filtered samples, fixed samples, took live samples. We went from 1.2 kilometers to 384 kilometers in all directions. We had collaborators from University of Alabama, Florida State, Gulf Coast Research Lab, Rutgers University, University of Oklahoma, Penn State University, University of Georgia, Scripps, and University of Tulsa. And if you go to this particular web page, which has been up since the very beginning on May 25th, you will see that you can see all of the data that were collected, pictures, the ships, daily reports from the vessels, everything's there. It's all public. So we have literally thousands of pictures, and the ocean does burn if it has this particular oil on it. Most of what we saw was a sheen at the surface. We also occasionally saw mousse, and we saw these droplets. This is at the drill rig at the leak closure, and they were siphoning off material at that point. We also took some sediment analyses, and we're analyzing those for what reached the shore and went in deep. We had a basic system of CTD, or conductivity, temperature, and depth. And this has a number of different bottles on it that can be fired at specific depths. It has a number of different in situ instruments. So for fluorescence, we had 2 different fluorescent detectors that would detect oil hydrocarbons. And so we could then go down all the way to the bottom and then come back and sample above and below the plume that we could detect, and then within the plume, and then at higher depths above that. At the surface, we started off with using the Halliburton bucket. That seemed appropriate in some ways. And you can see sometimes we could get some nasty amounts of emulsified oil and mousse. And here it shows some of the guys taking samples, and they're very close to the wellhead here, and taking the samples with and collecting the water, and then immediately filtering them and chemically fixing them and that sort of thing. Camilli had a paper come out on August 19th, just a couple of days before our paper came out. And this is the Woods Hole group. They were actually analyzing the deep plume with a number of different instruments and robots. And they could show bathymetrically that it was spreading out at about 1,100 meters. It's about the size of Manhattan, and but again, you couldn't see it or smell it. And he says that in his paper too. We found exactly the same thing that he did, and even though some of the press tried to say that we were somehow conflicting or that sort of thing, we weren't. We agree that basically our work is complementary. We sampled all around the wellhead and 2 different ships. This is the wellhead here, and going out in the southwest direction. And here you can see total N-alkanes in the concentration. We see this 1,100 meter, 1,200 meter high concentration. And at the surface, BTEX compounds, we also see this at around the same interval, though very little at the surface because immediately the BTEX compounds would evaporate. And then PAHs, we only saw those at the surface because that's the emulsified oil. We didn't see anything down deep. This is actually some of the more toxic components too. Now, just to give you a little orientation, the bugs can degrade oil. And in fact, by using oxygen and fertilizer, nitrogen and phosphorus, and proper temperature and pH, and that temperature could be cold, they can either produce carbon dioxide and water or daughter products. And the daughter products could still be toxic, so we're going to have some long-term studies that we need to do too. So down deep, we saw this definite plume with the fluorescence. The bacterial densities didn't seem to change all that much at 1 kilometer. Though I'll show you in a minute they actually were higher in density because they hadn't completely divided. They had gone up quite a bit higher at 5 kilometers. The oil started to go down, and at 10 kilometers it had gone down quite a bit. Densities were actually higher, and outside of the plume, no difference. Dissolved oxygen, we see a little bit of a blip at 1 kilometer. We see a major blip at 5 kilometers, and this is in the current, and a minor blip out here at 10 kilometers where a lot of the oil has been degraded. And again, none of that change outside of the plume. We did a number of statistical analyses. Phosphates were significantly lower in the plume. And here you can see the differences. It wasn't significant. Ammonia was significantly higher, not significantly higher. Nitrate was significantly lower. And dissolved inorganic carbon was not significantly different, but it was different and higher down deep in terms of the C13 ratio. Octadecane, all of the hydrocarbons were significantly different. The bacteria density was significantly different. Archaeo richness was not. Remember, this is an oxygenated environment, so there's no complete depletion of the oxygen. So there's no anaerobes down here. So, and a lot of those would be methanogens, that sort of thing. No significant difference, but the bacterial richness was significantly lower in the plume. So the bugs and both the bacterial richness and the total richness. So the bugs had decreased the number of species, but increased in density. So the dominant species were, as we'll see, oil degraders. POFA changes in terms of biomass were the same. The Oceanospirales clone, which was one of the dominant bugs that can degrade oil that we found, was significantly greater. Trans-cis ratios were not significantly different. That suggests that the bugs weren't under much stress, so they were basically fat and happy. And then we see particular lipids that we see in the membrane that are associated with oil, and those were significantly different too. Lipid signatures, biomass, you can see how different they are in terms of the average plume concentration versus non-plume concentration. You can see the community comparison shifts quite significantly too. We also used a phylochip, which is a 16S microarray. It has 50,000 different bacteria and archaea in one test. We could do this test in about 8 hours. And we found 951 subfamilies in all of the different samples that we took, but only 16 subfamilies were dominant, and mostly the proteobacteria, gamma proteobacteria, were enriched in the plume. So one particular group. We also saw aeromonads, Coluellaceae, just like the Santa Barbara study with Dave Valentine showed. And then also, of course, the Oceanospirales and a number of other species. And a lot of these are known to be either enriched by crude or can degrade oil hydrocarbons. We also did a direct comparison between the 16S and the phospholipids. And these, looking at all the different sites we had, they cluster together, and they're amazingly similar in terms of what we see. So in fact, this confirms with two completely different techniques that the community was quite a bit different and clustered together in the plume. We also did clone libraries, and we could see outside of the plume, only 5% of the clones were the Oceanospirales, but in two different samples, they were either 95% of the clones or 90% of the clones from within the plume. So that was another completely different test for dominant organisms. We also did pyrotag sequences, and this shows samples that came from outside the plume versus from within the plume, and they're all quite a bit different, and the diversity is quite a bit lower here. We also did community composition experiments, and this is with our pyrotag sequences, and you can see here again the community outside of the plume versus both of the plume samples that we did this analysis on. Now, we also did GeoChip, so a functional gene array. So we're looking at exactly how fast things respond in terms of enzymes being characterized. And so with this chip, which has, and we're using the GeoChip 4, which has genes for antibiotic resistance. These are highly conserved, so we don't care in this case what species is there, but do they have the gene, and was it highly expressed? Carbon fixation, carbon degradation, contaminant degradation. Of course, a lot of this was associated directly with hydrocarbons. When we did that and looked at particular genes that we know degrade the hydrocarbons and looked at alkanes, cycloalkanes, degradation genes, you can see they're all significantly higher and out in the plume versus the non-plume. And then even the aromatic carboxylic genes also were significantly higher out in the plume versus outside the plume. So both the alkanes and the pHs would be more degraded because of that. We could also directly show a direct correlation, and everything that's in bold here is significant in terms of different components that we could find at the oil at a particular site, and then the expression of that particular gene, how correlated it was. So you can see here that if there was a higher amount of that particular compound, there was more expression of that particular gene that could degrade that compound in the plume. And they responded fairly fast. As you'll see, going out 10 kilometers, it only took about 3 to 4 days to get out 10 kilometers. So this shows, at 1 kilometer, it shows the bugs. They've already started dividing, but they haven't completely divided, so I counted this as one bug. But actually, it probably should be counted as a number of different bugs. And then out at 10 kilometers, much higher densities, they're all fat and happy, completely divided, and that sort of thing. But we saw this long, incompletely divided bug, and which was the Ocean Aspergilles bug, close in. So we, a lot of these bugs are psychrophiles. They can degrade oil at 5 degrees or even 4 degrees. And they degrade it faster than at 20 degrees. So they're adapted to very cold environments. And so we took samples, live samples, back to the laboratory. And of course, normally you preserve these at 4 degrees, refrigerator temperatures. Well, when we got back to the lab, there was no oil there. But we found flocks. So we took and analyzed those flocks with the synchrotron FTIR, and we could see the proteins associated with the bacteria were in these flocks, the carbohydrates that are associated with the bacteria, and then small amounts of alkanes in the flocks, but also large amounts in these heat maps of oxidation products, nitration products, and sulfation products. So this is bacterial poop, okay? No easier way to say it. They had completely degraded it, okay? And all that was left was trace amounts of these alkanes and things. And it's like some of the stuff we see in sewage treatment plants and that sort of thing in terms of flocks and that sort of thing too. We also took water back from the surface water, much larger droplets, and here you can see an alkane droplet from the surface. And then we could show that there was a bacteria attacking that droplet. And we could show that there's an intense activity of the oxidation products in 2 different particular heat maps of different oxidation products associated with that bacteria attacking that droplet. So there's no doubt that these bugs were degrading that oil. And here you can in fact see the bacteria attacking the oil droplet. You may have seen this in some of the popular press, some of our pictures on this. And this is the oil droplet, and you can see the bacteria. Sorry, that's hard to see there, but you can see that fairly well in some of the other slides. So bacteria, marine bacteria, are known to completely degrade all of the components in the oil. However, they degrade some of it much more slowly. And we do have documented all the pathways for this, and this has been done over the last 30 years. So no surprises there. So what we did was to look at plume samples, and this is water that we collected and chemically analyzed for these particular alkanes, and the alkanes are 80% of the oil that was in the plume. That's why we focused on the alkanes. So we used a conservative estimate that it would take to go out 10 kilometers, it would take 2 days or 5 days. We also had data that was collected for BP by contractors, and it was analyzed by Louisiana State University and Lancaster Labs, and they directly sent the data to me to do this analysis, so I didn't look at BP analyzed data. We also did mixed consortia, and so this is a standard way to look for petroleum degraders at 5 degrees. So we used peptone with minimal salts, marine media, and went through a couple of incubations and inoculated it without peptone with the Macondo oil. We also took water from non-plume sites, set up these microcosms at 5 degrees, and inoculated it with Macondo oil. Actually, I like that the best, okay? Because it doesn't have some of the biases that some of the other things bring in. Range 1.2 to 6.1 half-lives, day half-lives. Pretty fast, but not unusual, okay? And that range is similar to what we see in other studies. We also looked at the alkane ratios, and that basically agreed with this. So as the shorter chain stuff is degraded, you see a change in the ratio of the longer chained alkanes to the shorter chained alkanes. And here we were using C26 to C15, C26 to C16, and C26 to C17. Close to the well, we don't see any changes. And then as you get out to 5 kilometers and 10 kilometers, we see a fairly dramatic change in that ratio. Now, the literature actually shows from our study, of course, we got 1.2 to 6.1 days. Our microcosms 2.2 to 3.5 days. However, with Prudhoe Bay oil, heavy crude, Venosa and Holder actually showed at 5 degrees they could get alkanes being degraded at 4.6 days half-life and 9.9 days without the Corexit. Cerrone did some studies in the St. Lawrence Seaway with PAHs, and he got 1.5 to 1.7 day half-lives. He did some other studies with particular components of the oil, again, very fast rates. And there was a Chinese study done with a Corexit-like compound, I think, and they were actually incubating at 10 degrees, and they got a half-life of 1.6 days. So this is not unusual, actually. So what happened to the oil? From July 27th to August 26th, we sampled 170 different sites, and we sampled all around the wellhead and then tried to intersect where the oil might have gone using transects and going out to 300 kilometers. We were below detectable limits in our CTD, which is less than 1 ppm. And for all of our chemical analyses and all these samples, we were below 2 parts per billion for all these samples. It was completely undetectable after July 27th for all our samples. It did not move out. It was completely gone as near as we could tell. We couldn't detect it anyway. So despite some of the, what you've heard and some of the people speculating where it went, this is actual data that we have, and also this has been released to the government too. Last Thursday, Kessler and Ballantyne and their cohorts published a paper about the methane. And you'll see me comment about that. The media had me comment about that. And in fact, they found the same sort of thing, that the methane degraded quite fast. You can see the oxidation rate constants. You can see the DO anomalies. And incidentally, the white dots that you see here are DO anomalies. So we see a slight dip in DO at those sites. So there's more flocks and more bacterial poop and that sort of thing at those sites. There was no oil left, undetectable. But we could see, in fact, that there was a remnant of it. So they saw their average DO anomaly and saw that immediately go away, and they couldn't detect anything after August for any of the methane, actually in July, late July, just like we had shown too. And this has some significant effects on climate, as they point out in their paper. So catastrophic releases of methane from the deep, potentially in areas that are adapted, all of that would be oxidized almost immediately before it reaches the surface. So it won't be methane that's going into the atmosphere. The most it'll be is CO2. So that's one analysis that may come out of this. The other thing that's good about this, and we have the same data, and he beat me to the punch, we basically also showed the isotopic changes. We also showed the methane disappearing, similar, almost exactly the same rates, and everything else. The good thing about the methane, too, is the methanotrophs can degrade over 300 different compounds. So almost all of the components in the oil could be fortuitously metabolized by the methanotrophs once the methane has disappeared, or by the methanotrophs. So we could be getting a deep cleaning effect, which may be good for the Gulf also. Corexit. So a lot of controversy about Corexit and its degradation. We haven't published this yet, but we looked at a lot of these compounds and how fast they were degraded. And they are degraded fairly slowly, especially the glycol compounds, the propylene glycol in particular. And we actually get faster overall rates at 4 degrees than we do at 25 degrees for almost everything in half-lives, 32 versus 62 days. So they do degrade much more slowly than the oil does. However, we never detected, and nobody else has ever detected it in the deep. So even though it was being injected at 10 gallons per minute, because it's water soluble, it immediately diluted out, and we couldn't detect it at all. That doesn't mean it's not there. Now, the propylene glycol, I have an interesting way of associating that for people, is that those of us that are older and that have had colonoscopies, the night before we've taken propylene glycol. And we know it's a really effective dispersant. And I hope, because I've had this done a couple of times, I hope that it's not toxic. At least as far as we know, it's not toxic. So, and that's a major component in the Corexit. It's degraded very slowly, but in fact, it is a major component. So what's left of the plume? We do see flocks, and we can record a lot of these. We have analyzed the sediment close to the flocks, close to the sediment-water interface, close to the wellhead. And we see, using the synchrotron FTIR, we see limited amounts of alkanes. And actually, this is so sensitive that it's basically still undetectable at 2 parts per billion. But we can see it in a heat map in the flock. For the fact that we're detecting these clay, carbonate, sulfate, orthosilicate, and luminate compounds suggests that this was drilling mud. So what we were seeing close to the wellhead, and remember, with the top kill, a lot of drilling mud was used. That's about all we see. We see a couple of places where we've seen little blips of oil because the oil was being incorporated with the drilling mud and then going to the bottom. I think in most cases, though, the oil was going to the surface in droplets. Now, dead zones. A lot of people want to talk about dead zones and the fact that they were worried that this may contribute to the dead zones. This was a paper that was just published in October, and this is from 2005. And basically, you can see what happened here in God, I can't read that number. In May versus July. And this is where the dead zones are. They're much closer to the coast. And actually, to give you an orientation, the Macondo well is over here, and the deep plume was moving in this direction. This is less than 700 meters in depth. So it's completely different, and that's why the government in our studies and Camille's studies and Valentine's studies suggest that, in fact, we don't think that the oil will contribute much to these dead zones at all. It's mostly the contribution is from these marshy areas and the Mississippi Delta coming around this in this direction. Now, natural seeps. This is from the Oil in the Sea 3, done in 2003 by the National Academy of Sciences. There's more seeps offshore going into the Gulf of Mexico than any other place in North America. The only place that comes relatively close or as significant is down here in Santa Barbara. And we know that from the tar balls and that sort of thing. Look at the amount that's here. Tremendous amount. So the equivalent of more than 400,000 barrels a year is going into the Gulf from natural seeps every year. And this has been going on for millions of years. So if I had to look for where I would find bugs that can degrade the oil down deep, this would have been the place, okay? In fact, we did find them. So it's not surprising, and it may mean that there could be some differences if other oil spills occur, the type of oil it is, and whether the bugs are adapted to this down deep at least. We may be okay in Santa Barbara, but we may not be in some other places that spills might occur. These are the pictures that we will never forget. And some people argue that this has become BP's logo. And certainly it was a devastating ecological catastrophe. I do not want any of you to take away the fact that it wasn't ecological catastrophe. There was a lot of oil out there from April 20th to July 15th. Tremendous amount of oil. However, let's look at some statistics, okay? 2, oil wells are all local. So in this case, we had 11 men, 6,104 birds, 605 sea turtles, 97 marine mammals were killed with the Deepwater Horizon spill, and it was 4.9 million barrels. This is as of the latest data that I have from the government, all the government sources on October 14th. The Casco-Busan spill right here in San Francisco killed 2,500 birds. That was minuscule compared to this, but it was very close to shore. The Exxon Valdez spill, 250,000 birds, 22 killer whales, 2,800 otters, and it was only 0.26 million barrels. It was very close to shore. It was in an area that wasn't adapted at all, and it was heavy oil, and it sank into the coastline, into the sediments. Major problem versus what we see with this. In the Moco Cadiz spill, 20,000 birds. That was also a light crude, 1.6 million barrels. The Ixtoc one, as near as we can tell, probably 10,000 birds were killed, but that's a very loose number. And that was 3.3 million barrels. Now this number could go up, but there is major differences in the amounts of oil and what effects potentially they'll have in terms of how quickly did we disperse that, how much went to the shoreline, how much exposure there was to the wildlife, etc. So, and I want you to know this is in the Houston Chronicle. My God. And so you can't escape the media, you know. So this came out in October 4th. A huge oil-eating microbe just ate Earl. And they got a drill rig in the background. So we are doing a number of different studies. We have about 15 papers that we have currently in preparation or submitted. We're looking at droplet size, modeling that, respirometers, shoreline, correxit biodegradation, metagenome. We've done single cell sequencing, which is a brand new technology. We have some great things that we're seeing on the metagenome, and hopefully that's going to be out soon. Surface plume biodegradation and community structure. We've done a lot of repeat sampling from where the oil was. We've done in situ hydrocarbon biodegradation mixing bug traps. So we put these out at different depths with at the riser and with Cephadec speeds, and I'll show some of that in a minute. Determine kinetics and rates of biodegradation under high pressure. Sediment sampling with oil in the corexit. And in the future, we want to look at natural seeps and see how the microbial structure compares and how it compares to this oil and the systems biology modeling. And then long-term studies on the marsh. So this shows the Cephadec speeds that we used. We put them in these bug traps, and they were put on the risers with the ROVs that BP had. And then our people put those out, or I should say my people put those out, and then they collected them and froze them and brought them back to the lab. And so with this, we have put the Macondo oil on these little beads, and then we can directly look at how fast the oil is being degraded as we pick these up at different time points. And then also see how the community structure changes. So we're going to get a wealth of information out of those, and that paper is being worked on right now on that data. We have also taken a number of different sediment cores, and you can see we've gone out over 300 kilometers in all directions, and where the plume might have gone and sampled below that, and have done a number of different cores. So each one of these sampling points we took 24 cores, and these are 6 inches in diameter and a meter and a half long. We froze them, sectioned them, and brought those back to the laboratory, and we're doing analysis on these. Most of what we see is we don't see a lot of oil in even the surface cloak, but you do see flocks. So we do see some evidence that there was some biodegradation, and we have to separate that out and see if it was really due to the oil or not. Thank you.
Okay, great. So we have some time for questions. Okay, Lynn, how about you?
Would you expect pressure to affect degradation rates?
So Doug Bartlett, I've been collaborating with him at Scripps, and he's one of the experts on barophilic organisms. He doesn't think that we'll see at this depth, at 1,500 meters even, that we'll see much effect from depth actually affecting the bugs at all. It's not till you get down to 2,500 meters or deeper that you see a significant effect, he claims. However, he did take samples. He's analyzing those now. I haven't seen the results yet.
Okay, all right, more questions. Okay, how about over there, and then we'll come back over here. Yeah,
Just at the very end of your talk, you were talking about the biology and saying, well, we had so many birds killed here and so many bird kills there. This is done in a very kind of inefficient sampling process. Right. I mean, it's a very inexact way of doing it, and it doesn't in any way comment on how it affected animals that maybe didn't turn up dead in the net of the survey and had other effects. So
I agree.
In your talk, you're sort of implying, well, this really isn't a big a thing at all, but it seems it's very premature to talk in any way biologic effects, or even comparing them, because they're done by different peoples using such inefficient metrics.
Yeah, there is a big difference, though, between all those different studies. Now, that's all I can say about that. Are those absolute numbers? We don't know and probably never will.
Never know.
Because it's just too difficult to make a lot of those estimates. We don't know long term if we might see attribute a lot more dead organisms in the Gulf. But for right now, what showed up was 6,100 birds.
Okay, and how many didn't show up?
We don't know.
I mean, it's a very inefficient sampling process.
Yeah. But 250,000 showed up on the Exxon Valdez spill. And they showed
I
up.
don't think they were counted as 250,000 discrete events. Somebody made an estimate by doing some sampling in certain areas and multiplying and taking a guess.
So I have to rely upon the government reports that have come out, and that's the numbers that we've gotten so far. There's long-term issues that we need to look at.
Okay, next question.
Is there any safe possibility of using the bacterial populations that you ran into here and in the Gulf for future possible oil spills up in North Alaska?
No, I don't believe in bioaugmentation for oil. We've tried it a bunch of different times. It hasn't worked for oil in marine environments. Usually within 72 hours, we see major increase in oil-degrading bacteria in almost any environment, in marine environments. That's been true of the 30 or 40 years of research that has been devoted to this. So what happens if we did take some of these bugs and try to grow them up? How would we get them down deep? It would be very difficult. What would we do in terms of growing them up under laboratory conditions? Because quite often what happens is that when we put those bugs back in other environment that they're not adapted to, they immediately die. So then they can become fertilizer for the indigenous bugs. But this is an extremely expensive way to make fertilizer. It'd be much better just to put fertilizer out there if there was that sort of nutrient limitation. We have seen iron increase the biodegradation rates, but this is typical of ocean environments, and I knew this. We didn't see any limitations in terms of nitrogen, phosphorus, or oxygen, so there's no reason to believe that we needed to do that down deep. Now, shoreline, it's going to be a different story. And that goes anaerobic, and it's going to be completely different, but we don't have enough data from that yet.
All right, we'll take one more question. Okay, how about right up here?
Where these bacteria fit into the ecology? Do other organisms eat them, or do they just die and sink to the bottom of the ocean?
Well, they'll be in those flocks, okay. Other organisms could eat them. Now, the thing that people a lot of times will say, well, so it could accumulate, the oil could accumulate in the food chain. No, because if you ate oil, if you degrade any of that oil, that's going to be incorporated into your cells and into the amino acids and lipids and that sort of thing that you normally make. You're going to excrete the rest of it. So we've never shown that the bacteria that use the oil as a carbon and energy source, that they can accumulate oil in the food chain. However, other organisms may be able to filter it and filter small amounts out and accumulate it that way, but not the bacteria.
I was thinking more along the lines of this being a very large carbon energy input. It's sort of like biomass.
Well, it's moving out to the southwest, so we don't think it'll have a major effect, though. The dead zone was a little bit larger, I've heard, but I haven't seen the data on that yet. The dead zone was a little bit larger this year, but that may have been due to just natural occurrences. Now, a lot of people have been looking for the oil along the shoreline, that sort of thing. It's almost impossible to find already. If you dig down, you can still find some of it in some places, but it's becoming much more difficult to find. It'll degrade very slow. If you saw on that one slide that I had, it was 62 days if you put it into sediment, the sweet Louisiana crude. Half-life is 62 days.
Okay, with that, I think we'll wrap up, and I'm sure Terry will be up here afterwards if you want to talk. So, thank you.
For more, please visit us at stanford.edu.